This visualization maps the leap from standard diagnostics to psychiatric analysis. A basic point-of-care test looks for a single binary indicator, a yes or no line for a pathogen. Psychiatric liquid biopsy expands outward into a multi-dimensional scatterplot, requiring precise measurement of multiple genetic variables simultaneously. Distinguishing schizophrenia from bipolar I today typically requires 1 to 3 years of longitudinal clinical observation. That subjective waiting period comes with an initial misdiagnosis rate approaching 70%. Executing this outside of a laboratory is an engineering bottleneck because the process requires precise relative quantification of highly fragile, low-abundance RNA transcripts. The proposed solution is the SZ-meter. This is a conceptual desktop point-of-care instrument architected entirely around automating a quantitative 18-gene diagnostic assay. Moving a psychiatric molecular test to a physician's desktop demands an uncompromising physical architecture. The hardware must perfectly execute delicate wet chemistry to yield data clean enough for a clinical diagnosis. The system operates under a strict biological input constraint. It requires a standardized 3 milliliter venous blood draw collected into a vacuum stabilization tube. A standard fingerstick capillary draw is actively prohibited. Uncontrolled red blood cell hemolysis, interstitial fluid dilution, and high variability in leukocyte subpopulations completely invalidate the relative expression math before the test even begins. Once drawn, the sample enters a single-use cyclic olefin polymer cartridge, flowing through the port directly into the primary lysis chamber. Upon entry, the sample undergoes rapid chemical violence. A guanidinium-based buffer lyses the whole blood, immediately denaturing the RNA enzymes that would otherwise destroy the target transcripts within seconds. With the cells broken open and the RNA exposed, the system initiates a solid phase extraction using silica magnetic beads to pull the target material out of the resulting lysate. The cartridge then runs rigorous fluidic wash steps over the magnetic beads. This strips away cellular proteins, genomic DNA, and specific biological PCR inhibitors like heme from red blood cells and immunoglobulin G. Finally, a low-salt elution releases the purified RNA from the silica beads, concentrating the target template down to a highly specific 10 to 20 microliter volume. This sequence of automated chemical lysis and magnetic separation exists entirely to protect the fragile transcriptomic signature from degradation and isolate it for quantification. The hardware must now take this microscopic volume of eluted RNA and route it cleanly across an 18-target reaction array, doing so within the footprint and a desktop footprint. Active mechanical pumps introduce high failure rates and cross-contamination risks. Instead, the instrument utilizes external pressure and flow controllers to pneumatically drive the fluid through elastomeric microvalves on the cartridge. A true point-of-care device also faces a severe logistical constraint. It must eliminate the requirement for refrigerated cold chain transport entirely. Liquid biological reagents degrade at room temperature. The instrument circumvents this by using dry chemical preservation for its target-specific primers and hydrolysis probes. These reagents are stabilized in a glassy matrix of trehalose and xanthan. They are pre-spotted and air-dried directly into the cartridge wells during manufacturing. The reverse transcription and hot start polymerase enzymes are preserved as lyophilized single-dose beads. They sit dormant in a reservoir, reconstituting only when the eluted sample physically washes over them. Combining advanced pressure microfluidics with glassy matrix reagent stabilization allows a highly complex molecular assay to sit shelf-stable in a clinical office for up to 12 months. All of that sample preparation is merely a precursor to the machine's primary physical challenge: performing quantitative real-time PCR thermocycling. The thermal stage must heat and cool the entire array rapidly, enforcing an extreme spatial uniformity of plus or minus 0.1 degrees Celsius across every single reaction well. This chart calculates the relative expression of the genes. The final value is determined by taking 1 plus the amplification efficiency, raised to the negative delta-delta CT. To find that exponent, the machine tracks real-time fluorescence in the target well to determine the threshold cycle, marked here as CT. That threshold cycle is then subtracted against an endogenous reference control well, producing the normalized delta CT value. The mathematical vulnerability lies right here in the efficiency variable, E. The calculation assumes that the exponential DNA amplification is perfectly uniform and hovers near 100%. If the physical thermal block is uneven, efficiency drops. A seemingly minor 5% efficiency drift compounding over 35 thermal cycles creates massive exponential error. Clinically, this minor drift distorts the target template estimate several-fold, completely altering the resulting data vector. When that distorted vector is fed into the device's locked classifier, the math will force the algorithm to flip the final psychiatric diagnosis entirely. The SC meter is a rigid physical thermal enforcement mechanism designed entirely to protect the mathematical integrity of the efficiency variable. Once the thermal cycles conclude, the firmware takes the stabilized, normalized expression vector and hands it off to a locked logistic regression model. This stored model maps the vector to a specific diagnosis by applying fixed mathematical coefficients to each of the 18 gene data points. The ultimate output is a calculated probability grouping, differentiating whether the blood signature belongs to a patient with schizophrenia, bipolar 1, or a healthy control. The machine must then render this differential probability into a user interface that a non-specialist can read easily at the bedside. The SC meter achieves this through a graduated dial interface. The indicator's physical position on the dial and the color of the region it lands in redundantly encode the most consistent diagnostic class. This specific graphical choice operates as a legal strategy. Patenting a user interface often triggers Alice Section 101 rejections for claiming a mere presentation of information. Utilizing a redundant position and color mechanism asserts a specific functional display method that is structurally patentable. The instrument's firmware successfully compresses billions of raw transcript measurements into a single probability dial, executing the translation through a legally defensible information architecture. A pristine hardware design does not automatically clear the path to market. There is a rigid intellectual property divide between hardware invention and biological ownership. The hardware creator holds the intellectual property for the desktop architecture, the microfluidic cartridge design, and the novel graphical user interface. However, the assay environment running inside that hardware, the precise combination of biomarker transcripts and the classifier coefficients, is entirely separate, pre-existing intellectual property. Under diagnostic patent law, natural human genes themselves cannot be owned, but the specific multi-gene panels, the quantification methodologies, and the logistic regression coefficients applied to them are heavily protected assets. This creates the IP divide. Securing a utility patent for a novel diagnostic machine does not grant freedom to operate regarding the specific biological panels it processes. For the SZ meter to function commercially, operating the proposed instrument requires independent licenses from Laguna Diagnostics, the patent holders of the underlying biomarker science. Before this device can be sold, the hardware engineering must be followed by rigorous assay bridging, analytical validation to prove it matches laboratory results, and comprehensive legal cross-licensing. Commercializing a point-of-care molecular diagnostic requires executing on two fronts simultaneously: engineering the absolute physical stability of the hardware and strategically securing the legal rights to the clinical truth it reveals.